Hello, my name is Shuan Zhang, a plant pathologist at the Tropical Research and Education Center, University of Florida in Homestead, Florida, the United States. Uh, Probably many of you know Florida, but some may not know TREC or Homestead in Florida. TREC is located in Homestead, about 30 miles southwest of Miami, Florida. Is the only state university research and education center focusing on tropical and subtropical fruits. As a plant pathologist, why am I talking about soil salinity? Soil salinity and plant disease occur commonly and frequently in the same field. Interactions between soil salinity and plant disease may influence agricultural production, impacting food quality and safety. Soil salinity is salt content in the soil due to soil salinization, a process of accumulating soluble salts in the soil. Soil salinity is often measured by electrical conductivity, EC, with a unit decisiemens per meter. Primarily, soil salinization is caused by saltwater intrusion on coastlines and excessive fertilizer application in inland areas. In Florida, in general, little information is available for the impacts of soil salinity on vegetable disease or crop disease. Soil salinity severely affects crop production in Florida. Most studies on salinity involve investigation of soil salinity effects on endangered tree species on the coastlines and also in the Florida Everglades as well. Only a few reports of soil salinity on citrus are available in Florida agriculture. Soil salinity either increase or induce crop disease. Varies with crop and disease studied. For example, soil salinity was reported to increase bacterial spot and late blight on tomato, fusarium wilt, and pythium damping off on cucumber. However, soil salinity was also reported to reduce powdery mildew on tomatoes. You are probably wondering why we study the effect of soil salinity on bacterial spot of tomato. Because bacterial spot of tomato is an economically important disease in Florida as well as the whole world. There are 4 species of Xanthomonas cause bacterial spot on tomato. In Florida, Xanthomonas perforans is the main species cause this disease. Bacterial spot is favored by warm and moist weather, which is common in Florida. Bacterial spot can cause significant losses to tomato growers. Dr. Qingchun Liu my former biological scientist initiated this project. He tested the effect of soil salinity on tomato plant growth in greenhouse and found that soil salinity at EC 3, 5, and 7 decimals per meter significantly reduced the tomato plant height compared to the untreated control. Also, he found these soil salinity treatments significantly reduced shoot fresh weight and shoot dry weight. My PhD student, Cassandra Pierre, conducted greenhouse and field trials to evaluate the impact of soil salinity on bacterial spot of tomato. So here is the results she got from the greenhouse test. The bacterial spot from the soil salinity treated plants was significantly lower than the untreated control. Here are the pictures showing the difference in bacterial spot on salinity treatments and also from the untreated control. You see there are a lot of layers of bacterial spot on the tomato plant leaves in the untreated control. However, the plants with the salinity treatment, EC3, has significantly reduced the disease, and EC5 even much lower, and EC7 only a few layers, almost no disease. Cassandra moved to the field to conduct field trials in 2022 and 2023. This is the picture of the field trials, and the results showed that soil salinity treatment at EC3, 5 significantly reduced the bacterial spot disease severity compared to the untreated control, and also significantly reduced the disease progress reflected by AUDPC, the area under the disease progress curve. So that's the pictures you see. The picture on right is the plants treated with soil salinity at EC5, and the left one is the untreated control. You see the treated plants were much greener compared to the untreated plants because of the reduced bacterial spot disease. So that's this slide shows the results from the field trial in 2023. The salinity treatment at EC3, 5, 7 significantly reduced the bacterial spot severity, disease severity, those green bars. And also for the AUDPC, which showed as the brown orange bars. So EC5, EC7 significantly reduced AUDPC compared to the untreated control. Interestingly, the effect of EC5, EC7 were equivalent to the mancoside, a couple bacteria site included in this trial as the group standard. So that's tomato yield from this trial, field trial. EC7 significantly increase the yield of extra large fruit in the first harvest compared to the untreated control. And also the yield, the total yield from harvest in first harvest significantly reduced the yield compared to the untreated control. And also the EC7 had the same, significantly increase the extra large fruit yield compared to the grower's standard mango size. But overall, the total fruit yield was not significantly affected by salinity treatment compared to the untreated control. Casera moved to study the possible mechanism, including the physiological changes and RNA-seq for transcriptomic profiling as well. This slide shows the results of the physiological changes in due to soil salinity treatment in inoculated plants and also non-inoculated plants. She tested the CO2 assimilation rate, transpiration, stomatal conductance. Overall, salinity treatments reduced all these three parameters compared to the untreated control, regardless in inoculated or non-inoculated plants. Here are the pictures of the symptoms on the tomato leaves from salinity treatment EC7. You see almost no bacterial spot lesions on upper side of the leaves or bottom side of the leaves. But you see from the untreated control, there were a lot of lesions of big bacterial spot lesions, also from the bottom side. So you see the significant difference between the treatment and the untreated control. Here is the data from the RNA-seq experiments. So this is the PCA, the principal component analysis. You see the genes profiles are nicely grouped together from the same treatment, like here now inoculated at 12 hours, but from different salinity treatment like EC7 and also control. And there are many different groups can be formed from the same treatment. So here are the DEGs, the differentially expressed genes detected from inoculated and non-inoculated plants when you compare EC7 versus control, untreated control. So there are a lot of more DEGs from inoculated treatment plants compared to the non-inoculated control plants. We are especially interested in DEGs in EC7 versus untreated control. So here are the key GG pathways detected from the EC7 versus untreated control. The main or significant pathways include plant hormone signal transduction, plant pathogen interaction, phenylpropanoid biosynthesis, MAPK signaling pathway, and ABC transporters. Interestingly, Cassera tasted fruit from salinity treatments and found that big difference in flavor from the salinity treated tomato fruits compared to the untreated control. So she invited 32 volunteers to form a panel to have a blind taste test. Data showed that aroma, sweetness, taste, and overall flavor quality were better in salinity treatments, EC5 and EC7, compared to untreated control. Then Cassera looked into the literature and found that there have been some reports on low, moderate levels of salinity treatment enhanced nutritional and flavor quality of tomato fruit. In summary, soil salinity significantly reduced the bacterial spot of tomato in our study and significantly improved flavor quality of tomato fruit. Soil salinity reduced CO2 assimilation rate, transpiration, and stomata conductance. Soil salinity reduces stomata conductance probably due to stomata closure, thereby reducing the entrance by the pathogen Pseudomonas perforans into the plants. Many DEGs were detected in soil salinity treatment, and also we detected many KGG pathways involved in soil salinity treatment, including plant pathogen interaction, phenylpropanoid biosynthesis, MAPK signaling pathway, plant hormone signal transduction, and ABC transporters. Future studies should be conducted to investigate further mechanisms in bacterial spot suppression and flavor improvement. To explore and develop strategies for reducing the effect of soil salinity and bacterial spot. Also, we have a couple of questions. How to manage bacterial spot under salinity influence? How to improve tomato flavor, fruit flavor, or quality by increasing soil salinity without compromising yield. Before I finish this talk, I would like to thank those who helped or assisted in this study, including those in my lab, Dr. Yi Huang, Dr. Pamela Dutra, and Yuanyuan Wang. Dr. Yingkong Li's lab, Guiqin Yu, Dr. Bruce Schaefer's lab, Anna Vargas, and also TRE's field crew who helped our field trials. I also want to thank Dr. Jeff Jones from the Department of Plant Pathology in Gainesville. the UFICBR Interdisciplinary Center for Biotechnology Research for RNA sequencing experiments. Finally, I want to thank USDA Agriculture Marketing Services through Florida Department Agriculture and Consumer Services Specialty Crop Block Grant Program to support this study. With that, I would like to take any questions you may have if we have enough time. Thank you for your attention.